All right. Good evening, everybody, and welcome to tonight's EEL webinar. Tonight we'll be having an extremely interesting and controversial topic, and we'll be discussing about the relationship between endometriosis and microbiota. As you got used to it, we'd like to thank our sponsor, Gedeon Richter, for their support for EEL webinar, and for that, please allow me to share a short video.
To make a medicine, you need to shift your perspective to see success comes from a series of innovations. Without the dedicated work of our highly trained researchers, no such progress would be possible. It is today that experts need to think about the diseases of tomorrow, and premium quality requires the latest technology. All these aspects come together to create new treatments that will improve the health of millions. This is what we work for every day. Gedeon Richter, health is our mission.
Okay, now it's my pleasure to present our tonight's moderator. Actually, he doesn't need any presentation. He's an EEL Executive Board member, Professor Attila Bokor from the Semmelweis University in Budapest, who's involved in so many projects, and he'll be moderating tonight's interesting topic. Attila, thank you for being here.
Thank you, Aline. Thank you for your kind words. And our guest today is Professor Paola Vigano. I think she's really an expert, an authority of the field. Everybody knows Professor Vigano. He has an very extensive work on endometriosis, and she's also presently the head of IVF center at the University Hospital in Milano. And she's also the co-editor-in-chief of the Journal of Endometriosis and Uterine Disorders, the official journal of SEUD. She has published over 250 articles, 250. I think this is very impressive. And she has more than 5,000 citations already. So she used to be also associated editor of Human Reproduction Update and Human Reproduction as well, the top journals in reproductive medicine. Professor Vigano very recently was recognized among the top 4 world experts in endometriosis by Expertscape ranking in Palo Alto. So I think she really doesn't need any more explanation. So I'm very happy that she accepted our invitation. Thank you, Professor Vigano. And could you please share your knowledge with us, is there any link between gut microbiota and endometriosis? I think this is really a very interesting question.
Thank you very much. Fantastic presentation. Thank you. Thank you for the kind invitation. I'm going to share my screen. Do you see my screen?
Yeah.
Okay. Can I start?
Yes, please.
Okay. So thank you very much.
No, I can't. Right now, I cannot see it.
Oh, sorry. Let me see. Okay.
Now it's perfect.
It's working.
Perfect. Okay. Thank you very much again for this kind invitation. So today we will, these are my conflicts of interest in the last 3 years. And today, the plan of this presentation is the following. First of all, we will talk a little bit about the general aspects of the microbiome. Then I will try to show you some breakthrough of the microbiome research in other fields, just to understand what is happening in other fields. Then we will talk about the limitation of microbiome studies, because I would like to communicate to you, to inform you about the limitation and to have a critical way to consider this study. Then we will talk about microbiome and endometriosis and the data. And finally, we will discuss about this very interesting study on Fusobacterium and endometriosis. And we will try to draw some conclusion. So about the general aspects of the microbiome. The interest in the microbiome has increased substantially in the last year, and this was due to the Human Microbiome Project, which was a US NIH initiative, which was set up to understand the microbiota involvement in human health and disease. And the project was launched in 2017 with 2 phases. The first phase was to characterize the microbiomes of healthy human subjects at 5 major body sites using 16S metagenomic sequencing. And then in 2014, the NIH launched the second phase of the project, which was called the Integrative Human Microbiome Project, whose goal was to characterize microbiome and host from 3 cohorts of microbiome-associated conditions using omics technology. The 3 conditions that the NIH wanted to study were pregnancy and preterm birth, with the aim to understand the microbiome changes during the gestational period and how microbiome could influence the neonatal microbiome. The mother could influence the neonatal. The second condition was inflammatory bowel disease, with the goal to understand how the gut microbiome can change in women, in adults and children suffering from inflammatory bowel disease. And the third condition was type 2 diabetes, to assess the shift in microbiome of patients compared to not affected individuals. Here you have this slide describes the biogeography of human microbiome. And if you observe the various composition of the bacteria in the various body sites, the bacteria are these in the legend, you can see that they are in different colors, and you can realize that, for instance, the Fusobacterium, which is here in orange, is substantially present in the oral cavity and not in the other district of the body. And everybody knows that the microbiota composition from the same site of different individuals are more similar than the microbiome composition of different sites of the same individual. Another example, for instance, here in violet, you have the Firmicutes phylum, which includes the lactobacilli, which is dominant in the vagina. Now we know that the microorganism and the host communicate in numerous ways, so including the production of metabolism and the modulation of the immune system, which is very important, but also through a direct exchange of biochemical and genetic material between the microorganism and the host. And in turn, the host can influence the microbiome in a variety of ways. For instance, in the gut, it can be the diet, but also the immune modulation, while in the vaginal, for instance, it can be the sexual habits. But how can we measure the microbiome? How can the microbiome be evaluated? So usually the genetic material is extracted from samples, for instance, fecal samples, and they are prepared for sequencing. And then the genetic sequencing are filtered and compared to a known database to identify to which microorganism that sequencing belongs to. And then from here, different multivariate analysis statistical analysis can be performed. So you can do an analysis of abundance that tells you how many percentage of the microbiome are made up of a specific organism. You can do an analysis of biodiversity that tells you what amount of individual bacteria from each of the bacterial species is present in your microbiome. But you can do also an analysis of function that can identify difference in metabolic function. And you can also do a principal component analysis that characterizes a sample and compare it to other samples. So, an extraction and different way to analyze the data. It is also important to underline that, of course, the microbiome and the microbiota are important, but also their metabolites are important. For instance, the gut metabolites, the gut microbiome seems to have an important role throughout the body, and it's very important to some aspects of women's health, because the microbiota of the gut consists of microorganisms that can produce an enzyme which is called beta-glucuronidase. And this enzyme can deconjugate estrogens when they were about to be excreted, causing them to re-enter into the body and remain active for all the function mediated by estrogen. And for instance, this is very important for the composition of the vaginal microbiome, because the vaginal microbiome is affected by the estrogen, because the estrogen can stimulate the production of glycogen. And glycogen in the vagina can promote the dominance of the lactobacilli. So I wanted to spend a few slides also to understand what is happening in another field, which is very important. And I choose as an example the study of the microbiome in the connection between the gut and the brain. Probably those of you that has participated to the West Congress, the World Endometriosis Conference in Edinburgh, may have seen how this system works, because there was a presentation there. But in summary, neuroscientists have a specific interest in this, in understanding the role of the gut microbiome for the brain. And there are two big challenges in the study of this interaction. One is to actually understand underpinning how the gut microbiome can send a signal to the brain. And second, since they have very interesting results on mice, they are trying to understand how much these interesting data in mice can be translated into humans. It's very important to underline that to gain evidence that the microbiome is involved in a system, a good strategy is to take out the microbiome from the system and see what happens. And this is the basis for the use of the germ-free animals to answer many questions of the microbiome. These germ-free mice have been studied a lot for the connection between the gut microbiome and the brain. But I want to focus the attention on this kind of mice because they have been also used in endometriosis. And just as an interesting point, these germ-free animals have been shown to live longer. So what the neuroscientists have brilliantly found, is that these germ-free animals, mice, have a variety of change in the brain, a lot of change. They have an hypermethylation in the prefrontal cortex of the brain, And particularly this feature is is a characteristic only of the male mice. Then these germ-free mice have a dysregulation of the neurogenesis, of the hippocampus neurogenesis. These mice have changes in a variety of neurotransmitters, including GABA and serotonin. They have changes in the immune cells in the brain. They don't function correctly in the absence of the microbiome. And finally, these germ-free animals have a disruption in the blood-brain barrier. So a lot of changes in this animal without the microbiome influence every aspect of this life of the mice, social life, but also all the functions that are based on the brain, which is an incredible demonstration. So let's go on a little bit on what are the limitations of this microbiome study. I want to underline that there are some limitations, not only in endometriosis, but in every field, that should be known to critically evaluate the studies on microbiome. And I want to share with you this fantastic paper published in Nature Biology that have listed all the myths and misconceptions on microbiome that everybody should know. First of all, one of the myths about the microbiome is that the microbiota outnumbers human cells by 10 to 1, which is not exactly correct. According to this paper, more detailed analysis indicate that the true figure, albeit still impressive, is probably closer to a ratio of 1 to 1. And it should be noted that the ratio is likely to vary from person to person and is dependent on factors such as the body size and the amount of fecal material that are present in the colon. So it is not as much as numerous as we thought at the beginning. Another misconception about the microbiome study is that sequencing is unbiased. Although sequence-based method has been transformative for microbiome research with this new omics approach, they are not perfect. Biases can be introduced at every step of sequence-based study, from sample collection to storage through laboratory-based steps such as DNA extraction to choice of the pipelines for the bioinformatics and to choice of the database used for the analysis. So the sequencing is not perfect, always perfect. And another myth about the microbiome, underlined by this fantastic paper in Nature Microbiology, is that most diseases are characterized by a pathobiome. We always say that disease is characterized by a dysbiosis, a pathobiome. And actually, these terms, dysbiosis, pathobiome, are simplistic. Because microorganisms and their metabolites are neither good or bad. They merely exist. Their impact on us as hosts are heavily dependent on context. Microorganisms or metabolites that are deleterious in one context cause not harm in another, and which is a very important point. And what I also would like to underline is that we have a lot of condition in the brain, the lungs, in the liver, in the intestine, and so on. What also systemic disease, and one common theme, and in this organ we can have a multiple sclerosis, Parkinson's disease, asthma, liver cancer, diabetes, etc. etc. But one common aspect among all these conditions is that they result in increased levels of inflammation, as you can see here. And such inflammation can in turn deplete the gut microbiota, reducing diversity and allow facultative anaerobies to increase, as you can see here in red. And so in the study of the microbiota, since all the conditions are associated with inflammation, it is very difficult to distinguish cause from effect in this study. And since we are talking about an inflammatory disease like endometriosis, what is true for all these inflammatory diseases can be relevant and important also for endometriosis. And I wanted to present also to share with you the results of this study, in which these authors are a group of authors that respond to a paper published in Nature. This big paper published in Nature found a stronger correlation. The paper in Nature is this one, and these are all the bacteria. And this paper published in Nature found a strong correlation between microbiome and 32 different cancer types. And they created a machine learning predictor with a near perfect accuracy and distinguish among the different cancer. Some months later, these authors, which are from this paper, and their results are here, check the sequencing of the Nature paper and found a strong error in the genome, leading to millions of false positive findings, largely because most of the sequencing identified as bacteria were indeed human. So they have concluded that the microbiome-based predictor for identifying these 33 cancer in the Nature study were completely wrong. So sorry for this long presentation on the general aspects of the microbiome, and we can go now inside into the data on endometriosis. And I told you that germ-free mice were used in the study of in the study of the microbiota. And actually these authors did this very nice paper, very good experiment, in which actually they have implemented a model using antibiotic-induced microbiota depleted mice. And I applied them in the field of endometriosis. So they surgically induce endometriosis in both controls and antibiotic and microbiota depleted mice. And they transplanted a piece of uterus into the peritoneum, and then 21 days later, they evaluated the lesion. And as you can see here, lesion were in normal mice were significantly larger and more vascularized than lesion present in the microbiota depleted mice. Then they did another model of endometriosis induction, because they injected endometrium into the peritoneum of both control and microbiota depleted mice. And again, they evaluated the lesion 21 days later. And again, also in this model of endometriosis, they found that the control mice had lesion larger and greater in number than lesion of microbiota depleted mice, supporting a role of microbiota in the development of the lesion. But then the authors would like to understand whether the gut microbiota was important for the endometriotic lesion growth. So to this aim, they injected the uterine fragment into the peritoneum of control and donor and microbiota depleted mice, and then transplanted the fecal samples from mice with and without endometriosis by oral cavage in these mice And then 21 days later, they evaluated the lesion. And what I have to say that here, when it's written NE, it's fecal samples from non-endometriosis mice, and here are samples from endometriosis mice. And you can see actually that fecal samples from mice without endometriosis, here, that you can see here, were significantly smaller, and they develop less lesion, a lower number of lesions compared to mice with fecal samples from mice with endometriosis. And this was a way to indicate that the fecal microbiota could improve to favor the lesion development. But then the authors ask themselves actually if actually it's only the microbiota depletion that in some way alter the uterine microbiome, with the consequence that transplanted uterine fragments in microbiota depleted mice are less able to form a lesion than fragments in control mice. So to test this possibility, the author injected endometrial fragment from vehicle and microbiota depleted mice in control mice and in peritoneal cavity control mice. And what they observe is that mice receiving the endometrial fragments, either from the vesicle or from microbiome depleted mice, develop lesion in similar number and volume and size. And so they concluded, the author concluded that the uterine microbiota might be dispensable, but not the gut microbiota for endometriotic lesion growth in mice. And the results of this very well done study is that microbiota depleted mice show reduced endometriotic lesion growth. The transplantation of gut microbiota by oral gavage of samples, fecal samples from mice with endometriosis, rescued the endometriotic lesion growth, supporting the role of the gut microbiota, while the uterine microbiota was shown to be dispensable for endometriotic lesion growth. Okay, fantastic data on mice. What are the evidence in human? We will see rapidly the paper in the vaginal microbiota in endometriosis women, in women with and without endometriosis, of the uterine microbiome and of the gut microbiome. So what about the vaginal microbiome in women with and without endometriosis? I didn't do a systematic review. What I did is taken the paper published on the vaginal microbiome in endometriosis, and I have checked all the bacteria that are present in the various increase or decrease if there is a dominant of some bacterium in the vagina of women with and without endometriosis, and you can see that the results are quite different. And apart from some concordance for coli and Shigella, the results are quite different among them, and I don't think we can conclude so far something about from this human data on the vagina. What about endometrial microbiome? Before showing you the results of the endometriosis patient, I want to underline the fact that the endometrial microbiome is receiving a lot of interest in the context of the reproductive medicine in the IVF, because there are some groups that has studied a lot the endometrial microbiome, and they found a good correlation between the endometrial microbiome and the results of the IVF outcome. This is just one of these studies in which the authors have investigated the reproductive impact of an altered endometrial microbiota in endometrial fluid on implantation, miscarriage, ongoing pregnancy, and live birth rate. And they have evaluated 35 women undergoing ICSI or side donation treatment with at least one good quality embryos. And then they classify the samples of the endometrial microbiome with 2 machine learning models. And both models provided similar conclusion that the percentage of lactobacilli in the endometrial microbiome might be a significantly variable, able to predict the live birth rate. And these are the results in which you can see very clearly that when the endometrial microbiome has a dominance of lactobacilli, you have a pregnancy rate of 70%, while when the endometrial microbiome lactobacilli is not dominant, the pregnancy rate slows down to 33%. What about endometriosis? Again, I didn't do a systematic review. I just checked the literature for the original papers. And you can see that there are some bacteria in endometrium, which is very interesting from a biological point of view, because endometrium should be a sterile environment. But when we check all the papers together, there are different abundance in different bacteria, and again, nothing is concordance, and so it is very difficult to do some results from this study. And what about the gut microbiome? Again, I check the papers. There are not a lot of paper on gut microbiome and endometriosis. It is interesting because on gut microbiome and endometriosis, there are more reviews than original articles. And but again, you can see that of course there are a lot of bacteria, but the different and the dominance of one over the others in patients with endometriosis compared to controls is absolutely not evident. So again, concluding something from this data is very difficult. We need more evidence. And I want to conclude with this fantastic paper on Fusobacterium and endometriosis, because it has been a paper that has gone to media, so it is a very, very, very good paper. The experimental, many experiments have been done, and now I will try to summarize, because it's a little bit complex to read the paper. So I would like to summarize the main point of this paper in order to clarify also the enormous amount of effort which is behind this paper. First of all, I would like to share the main table of this paper, which is the characteristic of the patient included. They including 76 patients without endometriosis and 79 women with endometriosis. They are all patient operated, and they are all patient hysterectomized. They have the uterus has been extracted, and these support the age, which is not very, which is over 40. You can see that both the age and the proliferative cycle of the women are at the borderline levels of the significance between the two groups. But the last is the results from the Fusobacterium presence in the endometrium of these women, and they have 7% in women without endometriosis and 64% in women with endometriosis. So, of course, highly statistically significant. So it seems that Fusobacterium infection is characteristic of women with endometriosis. And then what did the author do? The author did a lot of experiments, as I mentioned, and I would like to summarize them for you. So first of all, they did an expression profiling comparing between endometrial fibroblasts from patients with endometriosis, and fibroblasts from ovarian endometriosis, and also from fibroblasts of women with endometriosis, ectopic endometrium from women with endometriosis. And they found that the only genes that were confirmed at RT-qPCR level was transcaling. You can see here very clearly that the transcaling was highly expressed in fibroblasts from ovarian endometriomas and lower in the ectopic endometrial patient with endometriosis, and even lower in ectopic endometrial patient without endometriosis. Then they did a series of experiments in vitro in which they demonstrated that the upregulation of transcaling could promote fibroblast proliferation. But they also demonstrated transforming growth factor beta-1, that you all know that it's the most important molecule that allow the transformation from fibroblast to myofibroblast to start the fibrosis, can be induced by the expression of transcaling. Then they went to the animal model, and so they infected the syngenic donor mice with Fusobacterium in a model of endometriosis, and they injected the endometrium in the peritoneal cavity. And actually, they found that when the Fusobacterium, when the endometrium was infected with the Fusobacterium, the lesion that was formed in the recipient of the infected uterus was greater, bigger, and multiple compared to the when the endometrium was not infected by the Fusobacterium. Moreover, they could demonstrate abundant M2 macrophage infiltration, a higher expression of transforming growth factor beta-1, and transgranulin positive myofibroblast could be observed in endometrial stroma of uteri that have been infected with the Fusobacteria, and also in the endometriotic lesion, which was established by these infected endometrium into the mice. And even more interestingly, they treated the mice with antibiotics for a week, and Fusobacterial was no longer present in this mice. The M2 macrophage infiltration was reduced, transforming growth beta-1 expression was reduced, transgranulin expression was reduced. So they were all reduced in the endometrials or in the mice endometrium when they treated with the antibiotic. Moreover, the recipient mice that received the endometrium from the Fusobacterium infected samples that were treated with antibiotics developed significantly fewer endometriotic lesions than the control group without antibiotics. Here you can see, it's very summary what I'm telling you, and here you can see that these are the fibroblasts here in violet, and in orange is the Fusobacterium. When the Fusobacterium is present, you can have an upregulation of transforming growth factor beta-1 that start the fibrosis process, but also an increase in transcaling that favors proliferation, attachment, and mobility, while antibiotics can eliminate all these pathways. And I want to show you finally only one, this is a very clear and very simple experiment in which you can see that they have these donor mice, and they infected their endometrium with Fusobacterium, and 56% of these bacteria actually have an infection in the endometrium, while 44 no, but they use these mice, their uterine samples to have been inoculated into the recipient mice. And when recipient mice receive the infected or non-infected uteri, you can see that the number of the lesion and the weight of the lesion are significantly increased when the, uterus is infected by the Fusobacterium. So the conclusion of my talk are the following. We have very strong evidence for a role of endometrial and/or gut microbiome in endometriosis, but in animal models. In human studies are few. Usually they have a small sample size, and we are absolutely unaware on whether the microbiota dysfunction can be the cause of the consequence of the disease. We have absolutely, we need more evidence to understand the mechanism underlying the possibility to have a role in endometriosis in women with endometriosis on the microbiome. And I really would like you to understand that in evaluating this study, we based on what we have seen in another field, we have to be very critical on the collection and the sequencing bias that this study may have. And I would like to thank you for your attention.
Thank you very much. It was very interesting. So this is exactly what I expected, since I also read this paper on Fusobacterium. I didn't really understand everything. So I think it was very useful that you just summarized for us. And indeed, I think it's a good methodology, and it's a very, very solidly written paper. However, I'm not very sure that it will be very easy to reproduce it. So yes, but I don't want to say that it's bullshit. So I'm really looking forward for another group to re-evaluate this data.
Yes, so the point is that you may reproduce the animal models, the animal study, and you may reproduce the woman study. So there are two different points of view. So we need data. We need data absolutely in women because, okay, 76 women, but we need more data on that, absolutely. Because that women was hysterectomized women, and we actually, the paper didn't mention on how they collected that endometrium. So we don't know if they opened the uterus and took the samples, or rather they passed through the vagina and the cervix, which is not very good in the study of endometrial samples from the microbiome point of view.
Yes, definitely. Is there any question from the audience? Because we have quite a fairly high number of participants. Let me see. So there is one question. What is the up-to-date practical point of all that you have just said?
Nothing.
Maybe.
No. I would, for me, I would be very prudent because you have seen that the human studies are very heterogeneous. So I would stay there and see, and it's not so easy to tell to a patient to take antibiotics. We can have a resistance to antibiotics. So I would stay there waiting for other evidence. If they will for sure come other evidence.
Yes. So
what do you think, Attila?
In my opinion, I would be very surprised that if our patient just would take some antibiotics and all disease will disappear. So for me, and if it's true, I'm more than happy, because we don't do any more major surgeries on these patients. So I think this is a very bright future. But at the moment, as I understand, the results are quite conflicting, and there is no solid evidence supporting the role of microbiota on the formation of this disease and also the progression of this disease as well.
Absolutely.
And what you said, I think it's so true. And I'm still wondering that we have more review articles on microbiota than original ones. So this is a new era of meta-analysis. And for me, if you have a very nice meta-analysis published in a top journal, and it is based on very poor quality data, so your meta-analysis, it makes just no sense.
I agree totally. I agree totally. No,
So,
there are also a lot of narrative reviews, but I mean, it's an interesting topic, microbiome, for sure, but we need also solid data. Otherwise, we are talking about nothing.
yes. And are you planning any study on microbiota, Paola, in Milano?
We are in the phase to see if we can replicate the human study, because it's not so difficult. Because also they didn't the human study on Fusobacter, sorry,
Yes.
Fusobacterium. Fusobacterium. Because the Fusobacterium study has not been done with the sequencing. It was been done with an RT-PCR. So it's very easy to replicate but the problem is how to take this endometrium, because if you pass through the vagina, really the contamination is so possible.
It is.
Yeah, last day I was in another congress, and there was a colleague that was talking about an expert in microbiome that has evaluated our papers in OB gynecology. And this microbiome expert thought that it was crazy to think that passing the vagina and the cervix was nothing. Of course, it is important.
Yes, so there are a lot of things to clarify, but
Yes.
But anyways, I think this is still an interesting issue, because I also have the feeling that maybe diet has an impact on microbiome, and we see that some patients of ours, if they follow a diet, they just
Yes.
get somehow better. However, we have no evidence on the use of diet, so maybe diet and microbiome or gut microbiome, it has some link. So maybe in the future we will have some more interesting results, and maybe we can give a good answer to this question from the audience. Yeah. So if there are no more questions, would you like to say something for us? What is this beautiful picture? Where is this taken?
This place is on the Lake of Como, on the Como Lake, because I live between Como and Milan. So this is the place where I go on Sundays. Yes.
This is gorgeous.
Yes.
Very beautiful.
Yes.
So,
thank you very much for this kind invitation. Very much, really.
Thank you very much for your talk. It was very interesting, and we look forward to the new data, and hopefully we can have some more exciting discussion on the role of the microbiome and endometriosis. So thank you very much.
Thank you to you. Thank you.
Thank you. Bye-bye.
Thank you very much. Before closing the webinar, please allow me to share with you the upcoming EEL events. So next year in 2024, from the 6th until the 8th of June, we'll be having the European Endometriosis Congress in Bucharest. And until then, we'll be having 2 more robotic endometriosis surgery masterclasses. The first one will be on the 2nd and 3rd of November in Duisburg in Germany, and next year in March in Bordeaux in France. And again in December, Attila, you are going to host the next EEL masterclass in Budapest. And I think we can now call this an evening, and thank you all for being here and discussing this interesting and also controversial topic.
Thank you very much.
Have a nice evening, and
you too. Bye-bye.
Bye-bye.
Bye-bye. Thank you.